Hello everyone. Today's discussion is about plant roots. This lecture will expound on the different root systems, root modifications, primary and secondary growth of roots, and its internal structures. Let's begin. Roots are characterized as the subterranean organ of a plant. In a sense, this organ is mostly found beneath the ground and does not function for photosynthetic process, as differentiated versus the shoot system of plants. The roots, however, are responsible for the assimilation of nutrients from substrate, e.g., soil, organic matter, etc. The downward growth of the roots is mainly influenced by the gravitational pull. However, there are some modifications that are present in some species of plants, like the epiphytic roots of most orchids, though epiphytic roots are not mainly advantageous to assimilate nutrients directly from the soil. This modification is very important and vital as it supports the habit of most orchids that are heavily reliant on atmospheric moisture and nutrients that they assimilate from the barks of larger trees. They also provide unbeatable anchorage to this orchid species so as they remain attached on a branch or twig, defying the disturbance of strong winds. Sometimes, epiphytic roots may function for photosynthesis. Example, the leafless orchids under the genus Chiluschista, Tenophyllum, etc. Here shown in this slide is the Philippine endemic Thrixpermum integrum that you can find abundantly within UP Baguio campus hanging on pine tree branches. This orchid is endemic to the Philippines, and you can see here this is the leaf of the orchid, and these are the epiphytic roots of this plant. Types of roots according to origin: primary root, secondary root, or an adventitious root. Primary root originates at germination stage from the radicle of the seed, while secondary roots emanates from the primary root. It did not directly originate from the radicle. It creates a branching pattern of the root system. It is also relatively finer than the primary root. While adventitious roots are roots that form from any non-root tissue and are produced both during normal development, like crown roots on cereals or nodal roots on strawberries, and in response to stress, such as flooding, nutrient deprivation, and wounding. Shown in this slide is a root system which is composed of a primary root, sorry, okay. Shown in this slide is a root system that is composed of a primary root. Now from the primary root emanates secondary roots or the lateral roots. In some resources, they may use secondary, tertiary, or even quaternary roots, maybe referring to the extent of the branching in the root system. There are also root hairs, very fine roots, okay, where exchange of nutrients and water mostly occur. Also, root hairs increases the surface area for root substrate exchange subterraneally. Types of root systems. We have three main types of root systems. Okay, the taproot, the fibrous root, and the adventitious root, respectively. Let's look at the different attributes that are different among this root system. Okay, for the taproot, it is prominent in the dicot plants. The primary roots grow and become stout, and then for the secondary and tertiary roots, they grow directly from the primary root. Okay, it's more penetrating at the same time. Next is the fibrous root with prominent among the monocots. The roots develop from the lower nodes. They have the same length and diameter. They are very fine. They are very thin. Okay, they form matting most of the time, and there are other literatures that states that the production of the fine roots in the fibrous root system is due to the portion of the radicle that aborted during the early stage of the vegetative growth of the monocots. Okay, thereby allowing to protrude other fine secondary or sorry um, lateral branches directly from that point. Okay. While for adventitious roots, they develop from different organs like that of a leaf, stem, petiole, etc. Okay. There are also some resources that are saying that fibrous roots may actually be the same with adventitious root, but for the sake of clarity, we can just perhaps delineate adventitious roots as roots emanating from non-root areas like that of the leaf, stem, or even leaves. Okay. Okay. Adventitious roots in normal development. In the embryonic development, there are adventitious roots that may arise in a later period after the growth of the primary root. Of there can be immediately adventitious roots that may arise at internodes of plants during the early growth. Okay, so when the radicle starts to grow, there are already other roots that are forming not directly from the primary root, but maybe at the base, where there is a junction between the radicle and the shoot system of the plant. Okay, there are also embryonic adventitious roots that may arise from the internodes of plants. Okay, at post-embryonic development, roots may arise from internodes to provide additional mechanical support. Like that here shown is a corn, okay, producing adventitious roots from its internodes. Okay, these roots will soon form additional mechanical support for the growing upright plant through this, okay, brace or prop or stilt roots. Or in some species, nodal roots may arise during asexual reproduction like that of the seagrass, okay, nodal roots also, or even strawberries. Okay. Adventitious may also function to aid support for a plant through climbing roots, okay, emanating directly from the stem of the plant, or special functions like pneumatophores, okay, or the breathing roots that is common among mangrove species. But take a look at this one. The thing that's emanating directly from the stem of your mangrove species are the prop roots, okay, which adds mechanical support to the upright growth of the plant, while the roots emanating from the ground, okay, those are your prop roots or the breathing roots. So a mangrove species possesses two kinds of adventitious roots, prop roots and pneumatophores. In response to stress, adventitious roots help the plant regain anchorage or may function for the assimilation, especially when the original root system was already severed. Okay, so those plants that are product of margoting, grafting, or stem cutting techniques, they are all regarded as adventitious roots. So adventitious roots again may arise even if it's a dicot or a monocot. Okay, as long as it is not directly a product or a derivative of the radicle of your plant. Here are also some information about a taproot versus an adventitious root. I can just perhaps pause for a while. Maybe these are already self-explanatory, but I would like to reiterate some important characteristics of the taproot and the adventitious root, especially in terms of their penetration from the soil. In a way, the taproot travels deep into the soil, while adventitious roots are more of near surface only. Okay, so there develops from the radicle, develops from other areas, other organs, other structures of your plant. Moreover, taproots are more persistent compared to your short-lived adventitious roots. Okay, and so on and so forth. So, like the entire plant developing from a juvenile stage towards a mature state, roots also have two growth phases, the primary growth and the secondary growth. Here shown in slide is the primary growth of your roots. Now dissecting the primary growth of your root, okay, we have here a longitudinal section showing the different layers or zones across your developing root. We start from the base, okay? We have here a root cap, then upwards, the zone of cell division, zone of elongation, and then zone of cell differentiation. Roots gro- Root growth begins with seed germination. When the plant embryo emerges from the seed, the radicle of the embryo forms the root system. The tip of the root is protected by the root cap, a structure exclusive to roots, and unlike any other plant structure, the root cap is continuously replaced because it gets damaged easily as the root pushes through the soil. To aid in the friction, as the root penetrates the ground, it secretes a mucilage as a lubricant. Initially, roots grow in length by the activity of the root apical meristem, which is protected by the root cap. Proliferation and differentiation of the root apical meristem give cells a sense of organization, so as called as the first stage of root development, the primary root growth. And the primary root growth has some zonations. So let us first focus on the three main zones during the primary growth, okay, which is the zone of cell division, zone of elongation, and zone of cell differentiation or maturation. So as you can see here, divided by some arrows, okay, the different zones have an overlap. Thus, they don't have actually a clear-cut delineation between transition areas. Okay, so for the zone of cell division, it contains the apical meristem. It is the location immediately behind the root cap where cells are actively dividing via mitosis. So this area of your root, okay, in the primary growth is the site of active cell division. That's why it's called as the zone of cell division. Within this area of the root apical meristem are actively the cells are actually in mitosis, okay, but then there is a point where the cells a little bit halts in terms of their activity. This is called the quiescent center because you know in the cell cycle there are actually stages of rest, okay, so there in the middle right after or in the area where the apical meristem is found, okay, there is a quiescent center, okay, but on this portion these are all cells that are actively dividing. Immediately preceding the zone of cell division, you have your zone of cell elongation. By the name itself, it suggests that the cells are elongated. Okay? It is the location where the newly formed cells are growing or increasing in length, which in return adds length to the entirety of the root. This process requires uptake of water, which literally stretches the cells and increases their size. In return, it is responsible for the overall root elongation. Okay, so we expect that the cells here are rectangular, okay, in shape. And then lastly, the zone of cell differentiation. The thing about this zone is that prior to all the cells reaching the differentiation zone, they are totipotent, meaning to say that the cells doesn't have fate yet, or the cells don't have function yet at these different zones. Therefore, it is only when they reach or when they have already acquired, okay, enough nutrients, okay, or even at least be given fate within this portion, okay, that they are already differentiated. Differentiated in the sense that they have their fate, they have their function already. And you can only find this differentiated cell at this specific zone, okay. Moreover, the zone of cell maturation is the location where the newly elongated cells complete their differentiation into either a dermal, vascular, or ground tissue. Maturation is driven by changes in gene expression. Okay, so they're already expressing a certain gene. Either they will perform as an outer covering, they become an epidermis. If they will perform function for added surface area to increase assimilation of water and nutrients, they function as root hairs. Okay, or if they function to carry out, or they function for the assimilation of nutrients upward, okay, they may form the steel or the vascular tissue of your plant. If it's functioning for storage, then they may be a part of the cortex, and so on and so forth. So these are, or this is the primary growth of your root. Okay, detailed in this slide are also the areas where the primary meristems are located, viz. The apical meristem, okay, there, like I've been saying previously, the procambium, or the primary meristem of the vascular system, the ground meristem, okay, and the protoderm, the primary meristem that gives rise to the epidermis. Further, let us dissect the components of a root cap. In most cases, the root cap region has its own meristematic cell or apical cell. From the point of active cell division, it creates cells with rigid cell wall. Afterwards, the root cap cells start producing statoliths, Golgi apparatus, and mucilage secretion. The root cap portion that is in the periphery most of the time sheds off as a result of friction due to the downward growth of the developing root. Okay, the cells here shed off most of the time because of friction from the downward growth of the plant root. Hence, there is a need to create cells purposively from time to time. As the root system of plants is considered to be indeterminately growing, mean to say, we cannot assume a certain extent or length or girth of the plant root because it is continuously growing. But then it might a curiosity question might arise about how does the root know that it's growing or it is supposed to grow downwards. The answer lies in a specialized cell called statocytes, and these statocytes are found in the root tip, mainly concentrated in the region of the root cap. The statocytes contains highly polarized cells that contain starch-filled plastids. These are amyloplasts, also called statoliths, okay, here pointed by arrows, that sediment to the physical bottom upon plant reorientation within gravitational field. So a simple experiment to note or to check if these statoliths are really responsible for the downward growth of plant, okay, assumed that really these statoliths are responding to gravitational pull in a way that here, okay, there they have this root, okay, that is oriented sidewards. So in the cellular view, we can see the statoliths here, okay, that are also oriented the same way with, of course, the original orientation, which is supposed to be downwards. But then, as a response to gravity, these statoliths were pulled, okay, on this area, shifting a reorientation, okay, that will now trigger the downward growth of your roots, okay. So by this simple experiment, it was empirically concluded that your statoliths are really responsible to detect, okay, gravitational pull and also responsible for the downward growth of your plant root. Okay, this translucent area here is actually the root cap. Okay, you can see it's a little bit shiny. That's the mucilage secreted by your root cap. These are just some added slide about the internal structure of the roots. Okay. Note It is only the cells of the zone of maturation or differentiation that have already determined their fate or function. Below these zones, again, the cells are still totipotent, especially in the zone of active cell division. Thus, if we cut through cross-sectionally the zone of maturation or differentiation, we can already see their different cells and tissues performing different function. Okay, so we can have there a stele, the root hair, epidermis, endodermis, the pericycle, the phloem cortex, etc., which are not found within the areas of the zone of cell division and the zone of elongation. Okay, and as the plant matures, it becomes more extensive, it becomes more specialized. All right. Now, we know that plants are able to absorb water from the soil, but how exactly is this process being executed by the plant? The answer is through two main pathways, namely apoplastic and symplastic roots. And in combination with these two roots, okay, there is called the transmembrane root. Okay, so note it is only in the mature area of the root that water absorption can take place. Thus, much of the water absorption process happens in the area of secondary root growth. Now let's detail the two main roots. The apoplastic is sometimes called as the non-living root because it's a pathway which provides a route towards the vascular stele through free spaces and cell walls of the epidermis and the cortex. An additional apoplastic route that allows direct access to the xylem and phloem is along the margins of secondary roots. Secondary roots develop from the pericycle, a cell layer just inside the endodermis. This structure is characterized by the Casparian strip. Casparian strip is a suberized layer that forces all assimilated nutrients to move in the symplast in order to enter the vascular system. So note, though apoplastic root may take the area, okay, here within the adjoining cells, once it enters the area of the endodermis, there is a Casparian strip, okay, and the Casparian strip envelopes all, okay, the adjoining cell. Thus, water plus assimilated nutrients cannot directly penetrate the Casparian strip. Thus, from apoplastic root, it will go through symplastic root, okay, in order for it to cross the boundary of the Casparian strip. Now, in contrast, a symplastic root is sometimes called as the living root. Symplastic root provides a pathway to the vascular stele involving cell-to-cell transport by or through plasmodesmata. And these plasmodesmata are channels of cytoplasm lined by plasma membrane that transverse cell walls, and by these channels, no, allow the different nutrients and dissolved minerals plus nutrients to move from cell to cell without passing through the cell wall. So there is already the difference between the symplast and the apoplast. Apoplast is passing through the cell wall, while your symplast is passing through the interconnections plasmodesmata. Okay? Plasmodesmata represent direct cytoplasmic connections from one cell to the next. Right? There. And then lastly, the third pathway is called the transmembrane route, which involves the movement across cells and cell walls combining both symplastic and apoplastic route. Now, at some cases, there might be a more efficiency to proceed in an apoplastic route and then or symplastic route. And these two different routes of water absorption can be combined, and that's the transmembrane route. Okay, like for example, If the apoplastic root will proceed this way, and at this point of the cell, it may be inefficient for the plant to absorb water through apoplastic root, it may switch into symplast root. So from cell wall, it moves through the cell itself, then it goes through the plasmodesmata, and then when it reaches this point where the plant feels that it is again not efficient for it to proceed in a symplastic route, okay, it will or it can go back to an apoplastic route, okay. But the thing again I would like to emphasize is that when it goes here in the area of the endodermis, it should be all symplastic because of the presence of your suberized Casparian strip that is non-penetrable in the apoplastic route. Okay, right. Now, during continuous maturation, roots may soon develop lateral roots or secondary roots. Lateral roots are produced when the cells in the pericycle are ruptured. Okay, as you can see here, it's directly ruptured. The layer of the cells surrounding the central vascular cylinder begin to divide and form additional cell layers that push through the outer cell layers of the primary root and then ultimately organize a second root meristem. Note, the origin of the lateral root is designed in a way that it is continuous with the vascularization of the main root where it diverges. Okay, so in terms of origin, it's directly from the vascular tissue of your plant and it is not arising from your epidermis. So it's not epidermal in origin. Thus, lateral roots, yes, are not epidermal origin in contrast with the root hairs, which are early root trichomes that develops during seed germination. So when you try to grow a mango seed, it looks like there are hyphae surrounding the root, the radicle, right? So those are actually root hairs. Those are root trichomes and they are epidermal in nature. That's why they easily fall off when the plant matures. But in contrast, your lateral growth, okay, is directly emanating from the vascular tissue, and that is in terms of its origin, thus it doesn't easily fall off, making sure that the lateral root will have a direct extension or it's a direct extension of the vascular system of your plant. And I'm down to my last slide. Do roots turn woody at maturity? The answer is yes. Well, at least for dicot roots. However, in this slide, we can clearly see that the root, though woody as exhibited by the large xylems, as you can see here, okay, has a thin outer covering versus periderm in trunks. No? This is because a thicker outer covering will impede in the overall absorption process of the root. So imagine this is very thick periderm, no? So it will impede in the overall absorption process of the root. It will not make the root efficient anymore. So it should maintain a little safeguard of a layer of cells, but it will not make it very, very thick. Thank you.